Hi guys, welcome back to Unacademy NEET English channel. My name is Tina and I'm your biology educator. Guys, we have seen in PYQs that NTA is repeatedly asking questions around the scientists and their contribution. So let's quickly revise all the scientists for the final revision. Now, I'm going to use some flashcards which will help you revise all these scientists in a easy peasy manner. Starting with Carl Linnaeus, he is known as the father of taxonomy or father of systematics. He developed binomial nomenclature and he gave it all in his book Systema Naturae. Aristotle was the first person to give a scientific method of classification. He divided plants on the basis of morphological characters into trees, shrubs, and herbs, while animals were divided into enema and enema, that is on the basis of RBCs, animals with RBCs and without RBCs. Then we have the famous R.H. Whittaker, who gave us the five kingdom classification and introduced basically the kingdom fungi. Then Dmitry Ivansky, he's known for the discovery of virus. So the first virus to be discovered was tobacco mosaic virus TMV. But the name virus was never introduced by Ivansky. That contribution goes to Bejar Naik. Uh, he coined the term virus and explained virus as infectious living fluid, that is contagium vivum fluidum. He called virus is a venom, so that is how the word came. Then came W.M. Stanley. Just remember Stanley. Stanley was the first person to crystallize the viruses and demonstrate that majorly the virus consists of proteins. Theo Dinier, he discovered viroids. Now we know viroids are free RNA molecules and they are devoid of the outer capsid. And these viroids are known for causing a disease known as potato spindle tuber disease. Then Emden, Merhoff, and Paranus, EMP. And these three people together elucidated the glycolytic pathway. That is why glycolysis is also known as EMP pathway. Moving on to Hans Krebs. Hans Krebs is known for the discovery of Krebs cycle. He described the whole cyclic process of Krebs cycle, also known as TCA cycle or citric acid cycle. Then moving on to Charles Darwin and F. Darwin. That is F. Darwin, Francis Darwin, his son. So both son and father, they discovered auxins. Remember, it was discovered by father and son combination, Darwin and his son, and they studied the bending of the colleptile towards light, which is called phototropism. Now let me know in the comment section which plant did they study in and where they discovered auxins. But F.W. Wendt is known for isolating the auxins from the oat colleptile tips. That is, oat is also known as avena. Darwin discovered it, F.W. Wendt isolated it. So there should be no confusion. Now, he identified gibberellic acid and its role in causing the foolish seedling disease, also known as the Bakane disease. He saw that this gibberellic acid is being produced by a fungus known as Gibberella fujikuroi. That is where it was first discovered. Then came Skoog and Miller. Both of them, they are known for the discovery of cytokinins and their role of cytokinins in plant tissue culture. Then H. Cousins are known for H. Cousins is known for the discovery of ethylene, and he saw that plant ethylene is a plant ripening hormone. Now, Joseph Priestley, also known for the discovery of oxygen, he saw that air is essential for the plant growth. So, plants are actually improving the air quality, which is damaged by the animal like rat and burning candle. Jan Ingenhouse established that sunlight is required for photosynthesis. And do you know who gave the term photosynthesis? It was none other but Ingenhouse. Then came Engelmann. Engelmann is known for the discovery of first action spectrum of photosynthesis. Then Cornelius van Niel, he showed that oxygen is released from water and also glucose is stored in the form of starch. Then came Blackman. Blackman gave us law of limiting factors. That is, if a process depends on multiple factors, it is the least available factor which will determine the rate of the process. So the rate of process depends upon that factor which is found in the least quantity. Then our Robert Brown, he is known for the discovery of nucleus, not of cell. He is known for the discovery of nucleus. Discovery of cell was made by Robert Hooke, right? Okay, moving on to Matthias Schleiden and Schwann. Both were the German scientists. Remember that as per the new NCRT, German scientists who gave us the cell theory, who gave us the cell theory. Schleiden was a botanist and Schwann was a zoologist. Rudolf Virchow modified the cell theory and introduced another term to the cell theory. Virchow was the first person to describe that cells come from pre-existing cells, that is omnis cellula a cellulae. Then Camillo Golgi, as the name says, Camillo Golgi is known for the discovery of Golgi apparatus. Then George Palade, discovery of ribosomes. That is why ribosomes are also known as Palade particles. Now, moving on to Gregor Johann Mendel. Who doesn't know him? Mendel is the father of genetics. He studied monohybrid cross, dihybrid cross, and gave us laws of inheritance. And the plant he chose for his experiments was pea plant, that is Pisum sativum. Now, after Gregor's death, it was De Vries, Correns, and Sekmark who independently rediscovered the Mendel's work. Then Walter Sutton and Theodore Boveri, everything that Mendel studied on the allelic level, on the gene level, Sutton and Boveri studied it at the chromosome level and gave us the chromosome theory of inheritance. Then came Thomas Hunt Morgan, who gave the experimental verification of Sutton and Boveri's chromosomal theory. So, confirmed the chromosomal theory. And which organism were used by Thomas Hunt Morgan? Let me know in the comment section. Now, Alfred Sturtevant, the student of Thomas Hunt Morgan, is known for creating the genetic linkage maps, and he concluded that recombination frequency is equal to the distance between the genes. Recombination frequency is equal to the distance between the genes, and the unit that he chose for explaining the distance between the genes is centimorgan, in the name of his teacher, Centimorgan. Then Henkin was the first person to identify the sex chromosome, that is X body. Frederick Measure, known for discovering the DNA as nuclein in the cell nucleus. Erwin Chargaff gave us Chargaff's rules, which means purine is equal to pyrimidine. So amount of adenine will be exactly equal to the amount of thymine, while amount of G will be exactly equal to cytosine. These were the two basic rules, and thereafter. Then Hargobind Khurana is known for synthesizing the defined RNA sequences. That is, he discovered or deciphered, not discovered, he deciphered the genetic code. Along with Marshall Nirenberg, he developed a cell-free system for protein synthesis. Jacob and Monod, they discovered the lag operon model. Frederick Sanger developed the methods for DNA and protein sequencing. Then Alec Jeffrey is known for inventing the technique of DNA fingerprinting, while Louis Pasteur gave us the germ theory. Moving into the health and diseases now. Louis Pasteur was the first person who said that every disease has a germ which causes it. Also, he is known for his contribution in pasteurization technique, and also discovered rabies and anthrax vaccines. Then came Robert Koch, identified that disease causing bacteria like TB and cholera, that is why TB is also known as Koch's disease, and gave Koch's postulates. Fleming, Chain, Flore, uh, all three of them were given Nobel Prize. Fleming was the first person to discover the antibiotic, which is penicillin, but the true use of antibiotic, the true use of penicillin was mentioned by Chain and Flore, and we know that Chain and Flore used this antibiotic in treating the American soldiers during World War II. So that is why in 1945, all three of them were given the Nobel Prize. But the discovery was made by Alexander Fleming. Is that clear? And then we come on to Rosalind Franklin and Maurice Wilkins. These were the two people who were the main physicists behind X-ray diffraction images. X-ray diffraction images, based on which the DNA structure was deciphered. Right now, Watson and Crick basically used their X-ray diffraction images and proposed the DNA double helical structure. So the main structure that we study about DNA is the one given by Watson and Crick. Also, you must know that they introduced the secondary structure, that is the two-dimensional structure of DNA. Talking about Frederick Griffith. Griffith was the first person to demonstrate bacterial transformation, right? So Griffith is known for the discovery of genetic material. Discovery of genetic material. They never said it was DNA or that was not known, but the discovery of genetic material was made by Griffith, and they called genetic material as transforming principle, also as transforming principle. Now, the biochemical nature of transforming principle was not discovered by Griffith. Then Avery, MacLeod, and McCarthy, they identified DNA as a transforming principle. Then Alfred Hershey and Martha Chase confirmed that DNA is the genetic material using a phage that is a bacteriophage. Michelson and Stahl, let's understand. So, see, let me mention again, Avery, MacLeod, and McCarthy, they were the first ones to identify the biochemical nature of transforming principle was said by Avery, MacLeod, and McCarthy. Griffith discovered the transforming principle, but the biochemical nature was discovered by these three people. And Alfred Hershey and Martha Chase unequivocally concluded that DNA is the genetic material, and the organism used by Hershey and Chase was a bacteriophage, right? They used radio-labeled phosphorus and radio-labeled sulfur. Now, the organism used by Griffith was, let me know in the comment, and the hint is every MacLeod and McCarthy used the exact same setup. Now, talking about Michelson and Stahl, these two people proved that DNA replication is semi-conservative in nature. Now, this thing was experimentally proven by Taylor in case of eukaryotes. In eukaryotes, the semi-conservative replication method, so the organism used by Taylor and his team, Taylor et al., was faba beans. Is it clear? Let me know in the comment section who was responsible for discovering the semi-conservative nature in prokaryotes. Well, the hint is right in front of you, but still let me know in the comment section. Now, George Gamow was a physicist, but he is the main person to explain the triplet nature of our genetic code. So the codon is made up of three bases was discovered by George Gamow. Are these points clear? Now, talking about Ernest Mayr, known as the father of the 20th century, he gave the definition of the biological species. Catherine Issue, she discovered the curly top virus, which spreads this through a plant via a food. Now, she wrote a book known as Anatomy of Seed Plants, which is so famous that it is recognized as Webster's of plant biology. Then, talking about G. N. Ramachandran. Ramachandran is known for discovering the triple helical structure of collagen. Ramachandran explained this through conformation, and also he gave some conformations of protein through the Ramachandran plot. So remember him for the collagen structure and the Ramachandran plot. And what is Ramachandran plot? Ramachandran plot is important to study the conformations, that is structure of protein. Now, talking about Melvin Kelvin, important for he discovered the C3 cycle. That is why we call it Kelvin cycle. Now, along with J. Basham, this is clearly written in your introduction. Along with Basham, studied the reactions in the green plants forming sugar and other substances from the raw materials like CO2, water, and minerals by labeling the carbon dioxide with C14. So the pathway of C3 cycle was also studied as mentioned by Calvin. Now, Alfonso Cortai. Now, although Cortai was known for the discovery of organ of Cortai, which is a unit of hearing, the ear is not in your syllabus, so there are less likely chances that you will get this question, but let's not ignore. So, Cortai was the scientist who understood the structure of Cortai. This structure is present in the inner ear of cochlea, which is a structure for hearing. This is the unit of hearing. Now, Alexander von Humboldt, a naturalist and a geographer who gave us species area richness curve. That explanation that as we increase the area, the number of species also go on increasing, but only up to a certain limit, right? Was said by Alexander von Humboldt. David Tillman is known for his famous plot based experiments. He demonstrated that biodiversity stabilizes the ecosystem biomass. So he studied that those plots which had maximum diversity are the stable community. They form the stable community and undergoes less disturbance. Edward Wilson actually coined the term biodiversity. Moving on to Stanley Cohen and Herbert Boyer, they were the first to produce the recombinant DNA. Then Norman E. Borlaug, we know he is the father of Green Revolution and developed the semi-dwarf wheat. M.S. Swaminathan is the father of Green Revolution in India, right? Robert May estimated that global species diversity is around 7 million. Edward Wilson coined the term biodiversity, already seen. Already told you about Alexander von Humboldt. Then David Tillman studied. Frederick Griffith also gave us the transforming principle or the discovery of genetic material. So, guys, with this we complete the most important scientist around which the questions has been asked. Keep revising it once again and even before your exam. And all the very best.